Yeah, I'm from Revvity. Thank you for the introduction. So, as the introduction suggested, we're a big global life sciences company, but today's talk is actually only going to focus on a very small part of that. So if you want to know more about all of the genomics or next generation sequencing or other platforms or other things that Revvity do, please come and speak to us after my talk, because what I'm going to focus on today is the in vivo imaging side of Revvity's portfolio. So as a small introduction about myself, I've been with Revvity for 4 years now, just over 4 years. Before this, I was an academic. I've got a background in pharmacology and toxicology. I used to work at the University of Liverpool and research into regenerative medicine when I started doing in vivo imaging. And then I joined Revvity as an application scientist after this. So I'm going to start. This is going to be quite a high-level talk in terms of the content. So if you have any specific questions afterwards, you can come and speak to me in a bit more detail, but I'm going to keep it quite high level and try and go over the whole in vivo portfolio in kind of 15 minutes. So might be a bit of a whistle-stop tour, but we can speak in more detail afterwards. So in vivo imaging means in the living. So we're talking about imaging living animals, so whole animals, and we're generally talking about mice or rats imaging when we're talking about in vivo imaging. So one of the keys with in vivo imaging is that it allows us to image mice over time or rats over time, so we can kind of track the whole animal over time at many different time points. It might have been previously that we had lots of different groups that maybe received lots of different treatments, and we can only see what was going on at each of these time points when we cull the animal and we take the organs, we take the blood, we take the urine and run lots of tests. But imaging really allows us to see inside this kind of black box and understand what is going on in each of groups. And this is really a key concept with animal work, and it aligns to the 3 R's principle. So we want to reduce the amount of animals used in research, and we want to refine their use as well, and also replace them. But optical in vivo imaging really helps refine and reduce the amount of animals, so it does align really well with these principles. So I'm going to go over the 3 main in vivo imaging techniques that Revvity has in their in vivo portfolio, and that is optical imaging, that is micro CT, and that is ultrasound. So we're going to start with optical imaging, and optical imaging is just imaging light. So all we're doing is imaging light in the visible spectrum. So it's light that we could see with our eyes. If it were bright enough, it's often not bright enough, but it is just imaging light in the visible range. And we've known for a long time that light passes through tissue. If you hold your hand up to the sun or you shine a light behind your fingers, you will see light does pass through your fingers on your tissue, and this principle allows optical imaging to work, right? So and one of the key concepts in this is that tissue attenuates light, so the thicker the tissue, the less light will be able to pass through. And this is what makes mice ideal, because they're only a couple of centimeters thick, so we can have something giving off light in a mouse and we'll be able to detect this light. So usually we're talking about bioluminescence light, or we're talking about fluorescence light. I don't have really time to go into the details of these, but basically the key concept might be that we have a tumor cell perhaps that is expressing the enzyme that creates light in a firefly, so it will produce light when we give it we administer the substrate, and then we can detect this light. So we could track the progression of the tumor over time. More tumor cells, bigger tumor, more light to be produced. A reduction in tumor cells, less light to be produced. So that is one of the key concepts. So we can kind of track animals over time like this, and it's completely non-invasive. It doesn't harm the animal, it's just imaging them while they're anesthetized. I'm going to miss this in just to keep up with time. So we have a range of optical imaging systems, and these are called the IVIS systems. IVIS is in vivo imaging systems. So we have systems that are 2D systems, so the Lumina 3 and the Lumina S5. These can both do bioluminescence and fluorescence imaging in 2D. They can do spectral unmixing, which allows you to separate multiple fluorescent reporters inside the same animal. And they can image either 3 mice in the Lumina 3 or 5 mice in the Lumina S5. We also have these same systems which also have an integrated X-ray. So this allows you to do everything that the other systems do, but also overlay your images on an X-ray, which gives you a bit more anatomical information about where your signal is coming from. We also have 3D systems, which do everything the 2D systems do, but also allow 3D imaging of bioluminescence and fluorescence. And this allows you to actually put your bioluminescence or fluorescence signal in a more precise anatomical context, so we get some depth information as well in these 3D systems. And then the top of the range system is a 3D system with CT integrated, which is called the Spectrum CT2. This does everything the 3D system does, but it also has an integrated CT, which is a 3D X-ray, right? So we can overlay our three-dimensional bioluminescence or fluorescence images with a three-dimensional X-ray, so we can really see exactly where these signals are coming from. So these are the kind of images we see with these systems. So 2D would be a photograph with your bioluminescence or fluorescence signal overlaid on top of it. These 2D ones of X-ray give you a bit more anatomical information, so we can start to see the skeletal structure and maybe some soft tissue. 3D systems, we could start really seeing how deep these signals were, so perhaps how deep the tumor was or deep the metastases were in these animals. And then the 3D systems with CT, really a precise anatomical reference with your animals. So these IVIS systems have been around for over 25 years now, and we are the market leader in optical imaging, so over 25 years and over 30,000 peer-reviewed publications have used these IVIS systems. So yeah, if anyone has done any optical imaging or heard of IVIS systems, there's a very good chance that you've used one of these systems in the past. So in terms of application areas, it's very versatile system. The most important thing is that you can create a reporter that will emit light that you can detect. So I've mentioned tumors already, but you can do infectious diseases, you can create viruses. The COVID virus, obviously a few years ago, was a huge area of research. You can modify the virus so it will insert a light giving fluorophore or a light emitting enzyme that will code for that as well. You can do stem cell research, which was my area, neuroscience, cardiovascular, gene therapy, information. It just depends on creating an appropriate reporter that will emit light in these animals. So yeah, this slide just goes over some of these different application areas, but very vast. It's all just based on creating a nice reporter for your area of research. Okay, so that was a quick tour of optical imaging. So next we're going to talk about micro CT imaging. So micro CT imaging is, well, it stands for microcomputed tomography, and as I've mentioned before, a CT image is effectively a 3D X-ray. So if we think of a clinical X-ray you might have if you broke a bone, for example, that is a planar image, so just one orientation, and a CT is basically many of these taken at 360 degrees around the subject, and then you reconstruct a 3D volume of this. So that is what these systems do. It's the exact same technology that's used clinically, so you have clinical CT scanners as well. Obviously ours is not as big as the clinical ones, but the whole principle is based on how strongly different tissues or organs attenuate these X-rays. So bones are very dense, they attenuate a lot of X-rays, whereas soft tissues are less dense, so X-rays are able to pass through, and this allows us to generate our images. So yeah, this is a clinical CT system here at the top. It's the same principle in our system, it's just much smaller, but the mouse stays still, it's asleep, and the X-ray detector and the X-ray source rotate around the animal in 360 degrees. And we use this to build our images. We can do really nice high resolution images of bone, but we can also image soft tissues too. So we can do lung acquisitions, we can do cardiac acquisitions, we can gate for respiration, we can gate for cardiac cycles as well to really get a lot of information on cardiac function and lung function as well. So we have the Quantum GX3 system. It's a fully shielded system, so although it uses X-rays, there's no risk of X-ray exposure to the users. It's a small footprint, so it's probably about this by this. It fits nicely in a room. I should say, you guys have got an IVIS Spectrum and a Quantum system just down there. So there is two of these systems right here. It can image a range of different fields of view, so we can image from tiny ex vivo samples, so mouse bones, for example, in non-living mice, obviously, but we can go all the way up to mice, to rats, to rabbits, to small cats probably, or ferrets in this system as well. It's high speed, so we can do a full rotation in 4 seconds, but obviously the longer the scans, the higher quality of the scans as well, so it's completely variable. We can do functional imaging by doing gated acquisitions, which I mentioned earlier. So yeah, we can gate for both the cardiac cycle to get imaging peak diastole and peak systole of the heart to really tell us about how the heart is functioning, and we can get peak inspiration, peak expiration to really tell us about how the lungs are functioning, and everything is reconstructed on board the system. So yeah, if you think of X-rays or CT, you probably think of bones, but it's not just bones. We can do dentistry, we can look at the structures inside the bone, so we have the resolution to kind of look at the trabecular structure. So we can look at oncological applications, we can look at pulmonology, we can look at cardiology, and we look at endocrinology as well. So we can look at kind of fat, body composition, fat amounts in mice. So lots of different application areas as well. And we can actually even co-register data from the micro CT with data from the IVIS systems that I mentioned earlier. So if we do a 3D acquisition for a bioluminescent signal with an IVIS spectrum, we can then take the data from this and co-register it with our data from our micro CT as well. So we can kind of get a really high resolution CT on top of a really precise anatomical location of your bioluminescent tumor, for example. So the last modality I'm going to talk about is ultrasound. So that's with our Vega and VivoJect ultrasound systems. But a little introduction to ultrasound. If you think of ultrasound, you might think of scans you might have for pregnancy, and it's the exact same technology. We are measuring high frequency sound, ultrasound, with a transducer, and it's exactly the same technique used in the clinic, but with our system, you do not touch a transducer. It's fully automated, it's hands-free, and it's robotic. So we have these kind of bays on top of the system that the mouse just lies on, and the mouse is asleep again. We just need a few drops of water to kind of couple the mouse to this membrane. We draw a region of interest around the area of the mouse we'd like to image. We click go, and this transducer will sweep along the underside of the animal and build a 3D wide field volume of the animal. So the fact that you don't need to touch a transducer makes it extremely easy to use. You don't need a dedicated ultrasonographer to run this for you, and the free imaging bays makes it high throughput as well. So these are the types of images that can be generated with the ultrasound. So if you're familiar with ultrasound at all, usually you kind of see an axial slice across this way, which can be quite difficult to interpret if you're not familiar with ultrasound. Whereas with the Vega, we show a coronal slice. And hopefully you can appreciate here that we've got the spine running down the back of the mouse, we've got the ribs up here, left and right kidneys here. This dark area here is a tumor actually in this mouse. But yeah, it's much easier to interpret than classical ultrasound. And this type of image takes less than 2 minutes to acquire. So it's not just these kind of 3D volumes we can create. We can also measure fibrosis. So we've got a mode that we call shear wave elastography. And this again, if you go to the clinic to have your liver scanned, if they suspect you might have a liver fibrosis, you would have this exact same technology in the clinic as well. Sometimes it's called a fibroscan, but we introduce a shear wave into the liver with the transducers. And if you think of a stone being dropped into a lake and you see the ripples propagating outwards, this happens with a sound wave in the liver, and we measure the speed of this sound wave outwards, and this can tell us about how stiff the liver is or how fibrotic the liver is. So yeah, this is completely clinically translatable, and it's all correlated to actual stiffness values in the liver. We can also assess vascularization, so this might be good if you're studying tumor vascularization. We can look at how the blood vessel supply to the blood supply to the tumor is changing over time, both morphologically and more quantitatively the density. And this is with a contrast agent called microbubbles that we have to infuse into the animal. Lastly, on the Vega system, is this going to play? Yes. So we have this peripheral accessory which allows ultrasound guided injections into animals. So this is quite a common application for ultrasound, in that people can use the ultrasound transducers to guide injections into certain organs or tissues in the animal. So this might negate the need for surgery. In the past, you might have had to kind of open the mouse to inject your treatment or cells into a specific location, whereas now we can put the mouse to sleep and just inject it minimally invasively. So this video is showing an injection into the left ventricle of the heart. It's all controlled robotically, and yeah, it's stereotactic frames which can do lots of different angles to allow injections into lots of different tissues and organs. And it's a heated stage as well, so the mouse will maintain its body temperature while it's being imaged. And that just sits alongside the main Vega ultrasound system. This slide is just showing lots of different applications which is possible with the Vega. So tumor volumes, tumor vascularization, we can look at fatty liver deposition, we can look at liver stiffness, liver fibrosis, kidney vascularization, pregnancy obviously. We can also look at cardiac function, so we can get lots of information on how the heart is functioning as well with the ultrasound. In the last minute or so, I'm just going to introduce a new software that's just been released in the last, well, in 2026 this year, and this is a AI assisted analysis software. So I'm sure you guys have heard a lot about AI in recent years. So Revvity have just introduced this living image synergy AI software, and this is an analysis software. It doesn't replace any of the existing softwares we use to acquire data. It's just designed to speed up and streamline the analysis part of the workflow. If anyone has done any imaging, you're probably aware that acquiring the data itself can be quite straightforward, but then you come to analyze it and it can involve a lot of segmentation and time-consuming analysis. So this aims to speed that up. So we have AI-assisted tools to kind of automatically segment organs or whole bodies of the animal, and I think I've got a video which shows this. So yeah, this is some IVIS data, so optical imaging data over different days. So it might be a tumor growing, and we can ask the system to automatically segment the mice, the bodies of the mice, and it will automatically do this, and then it can plot this data in a graph for you. So this is actually showing the change in the light output from a tumor over time. And yeah, it's AI guided. We're not asking you to blindly trust what the AI tells you. We always want a human in the loop to kind of ensure that things are going as they should, but the software will do it automatically and it will plot the data for you. Very last thing, I promise. So we also have a range of imaging reagents as well for our in vivo and Vega ultrasound applications. So lots of different fluorescent probes for different disease states, lots of different bioluminescent probes for transfecting cells to express 5-fluoroluciferase and different fluorescent probes, bioluminescent bacterial strains, lots of bioluminescent tumor cell lines, and microbubble contrast agents for the ultrasound system as well. And yeah, this just summarizes what I've just said, but please come and speak to us if you have any questions, and I'm obviously happy to answer some questions here as well. So thank you very much for your attention.